there, this is FemmeSciComm's 12th vlog. I was on a brief hiatus, but I'm back now. I wanted to reconfigure my posts a little bit, and I had to recuperate from the cold that I got when I was in Los Angeles. And some good news, I'll be moving back to Los Angeles sometime soon. Lighting over there is just a bit easier to deal with than here in Chicago, where it's darkness 20 hours out of the day. My video today is on endometriosis, and this paper was recently published in Endocrinology, Genomic Function of Estrogen Receptor Beta in Endometriosis. The first author is Sang Han, and the senior author is Bert O'Malley, both from Baylor College of Medicine. This study tries to answer what is the genomic role of estrogen receptor beta in endometriosis. It's not a surprise that this reproductive disorder is estrogen dependent. Steroid hormones are super important in the female reproductive tract, and any change in that balance can cause cells to act abnormally. Estrogen is a small steroid hormone. It usually binds to estrogen receptors to enact its function in a cell. There are two main types of estrogen receptors, estrogen receptor alpha and estrogen receptor beta. In normal endometrium, ER alpha is expressed at normal levels and ER beta is barely expressed. However, in endometriotic tissue, ER beta is highly expressed and via several research studies decreases or represses the expression of ER alpha. So this paper wants to find out what exactly ER beta is doing in the endometrium and endometriotic lesions. First, the researchers established a mouse model where they overexpress estrogen receptor beta. And similar to what happens physiologically in humans, ER beta overexpression led to large lesions outside of the uterus. Then the researchers took endometrial and endometriotic tissue out of the mice to test the expression of all their genes via RNA sequencing. They also wanted to identify which genes ER beta physically binds to in these tissues via chip sequencing. First, we'll look at the pathways and genes that are affected with the ER beta overexpression in the utopic endometrium. By integrating their RNA-seq and chip-seq data through a bioinformatics software called Beta, they found that 2,082 of 19,862, or 10.5% of genes, are both bound by and upregulated due to ER beta overexpression in utopic endometrium. ER beta was found to upregulate pathways such as E2F and MYC, which are associated with cell proliferation. ER beta also binds and downregulates 2,513 of 19,862, or 12.7% of genes in the utopic endometrium. TNF-alpha and NF-kappa B signaling genes are downregulated, and these pathways have been implicated in implantation and successful pregnancy. Now we'll look at which pathways and genes are affected in the ectopic lesions with the ER beta overexpression. Again, by integrating their RNA-seq and chip-seq data, they found that 2,001 of 16,277, or 12.3% of genes, are both bound by and upregulated due to ER beta overexpression in ectopic lesions. G2M transition and IL-6 JAK-STAT3 signaling are associated with elevated proliferation activity. Genes involved in reactive oxygen species were elevated, suggesting that ER beta regulates ROS signaling for the survival of ectopic lesions. ER beta also binds and downregulates 1,696 of 16,277, or 10.4% of genes in ectopic lesions. The downregulation of interferon alpha signaling in ectopic lesions is associated with endometriosis progression. The researchers suggest that ER beta directly suppresses interferon alpha signaling in ectopic lesions to promote their survival. So what does this all mean? This research group determined what ER beta is doing in the utopic endometrium and the ectopic endometriotic lesions of female mice with endometriosis. They combined what genes ER beta regulates, as well as which genomic regions ER beta physically binds to, in order to determine what overall pathways ER beta is altering in endometriosis. There are caveats and factors to keep in mind. This is a mouse model of endometriosis. Mice do not menstruate but have an estrous cycle, so we don't know if these results can be recapitulated in human women. Next, ER beta is not normal ER beta, but ER beta with a flag tag to it. We don't know if this flag tagged ER beta is acting like the normal ER beta we find in women with endometriosis. Also, functional studies are needed to test if ER beta downstream genes cause endometrial dysfunction and endometriosis progression. Overall, this study starts to unravel the mechanism behind endometriosis progression through ER beta. This study confirms the use of birth control or hormonal therapy in regulating this reproductive disorder because it is highly estrogen dependent. In the future, it would be helpful to identify specific downstream genes that cause endometrial dysfunction and endometriosis progression in order to use precision individualized therapies for women with endometriosis that don't want their fertility affected or that don't want to have to cope with the adverse side effects of hormonal therapy. Thank you for reading, listening to, or watching my assessment of this paper on endometriosis. Keep informed, stay interested, and keep doing great women's health research. Thank you for watching. If you're interested in this content, make sure to subscribe to my channel. And you can also check out my website, FemmeSciComm.com. And you can follow me on Facebook, Twitter, Instagram, and Pinterest at FemmeSciComm. Bye!